And what's wrong with this drawing is that the wrong homologs are paired. The big dad chromosome should be paired with the big mom chromosome, but it's still paired with a little dad chromosome. And the big mom chromosome is paired with one of the dad chromosomes, but it's not the right one. So the daughter chromosomes are definitely going to get the wrong sets, incomplete, mixed up sets of chromosomes if this meiosis went to completion. Okay, here's a little simulation of meiosis just to remind you of how it works. I've got 4 chromosomes here, 2 homologs of a short chromosome and 2 homologs of a longer chromosome. The first thing that's going to happen is that the DNA is going to replicate. Sorry, red homologs have replicated, and our orange homologs have replicated. Now the homologs are going to find their partners, and they do this just by randomly moving around the cell. We'll discuss later exactly how this happens. Eventually, they find their partners and align along their whole length. Once they've aligned, they're pulled by spindle fibers in a kind of a tug-of-war that gradually moves them to the center of the cell. And once all the chromosomes are paired and at the center of the cell, they're pulled apart into 2 daughter cells, and the cell divides. Now let's go back to our flat 2-dimensional drawings of meiosis. The first steps are, after DNA replication, the sister chromatids stay together just like in mitosis, and then the homologs are brought together and locked side by side. And this allows the homologs to be pulled apart by the spindle fibers. 1 homolog to each daughter cell. But how do they find each other? How do they line up correctly? So here's another problem. The answer is that they find each other and line up correctly and are held together by base pairing. Remember base pairing? We talked about base pairing in Module 1 and in Module 2 when we talked about DNA replication and mutation. But base pairing is back because it plays an essential role in meiosis. So base pairing is how the homologs find their partners, how they remain precisely aligned. So to help you think about this, I want you to look at this other wrong drawing of chromosomes in meiosis 1. What's wrong here?